This protocol demonstrates a straightforward in vitro genetic modification methods for the molecular study of the primary cilium-dependent signaling pathway in the primary granule cell precursor cultures. Due to the cost, low fiability, and poor efficiency of the current transfection method, we introduced a simple, cost-effective, and efficient electroporation technique for investigating cilium-dependent signaling pathway in primary GCP cultures.
To begin, add 0.5 milliliters of culture medium into each well of the 24-well culture plate containing coated cover slips, and keep it warm at 37 degrees Celsius in a carbon dioxide incubator. Pipette the required number of cells into a sterile 1.5 milliliter microcentrifuge tube and spin at 200 times G for 5 minutes at room temperature. Discard the supernatant and resuspend the cell pellet in 200 microliters of Opti-MEM. Repeat this procedure twice to ensure no residual culture medium is present in the tube. Set the parameters of electroporation as listed. Pipette the electroporation reaction gently to mix well, and use a long P200 pipette tip to transfer an exact volume of 100 microliters of the mixture into the 2 mm gap cuvette. Once the cuvette is inside the cuvette chamber, press the omega button of the electroporator and note the impedance value by adhering to a precise volume of 100 microliters. The range of impedance values should be approximately 30 and 35. Press the start button to initiate the pulse. Record the measured current values in joules shown on the reading frame.